Welcome everybody to this webinar organized by CosmoBio and Lexogen. My name is Martina Sauert and I'm a product manager at Lexogen, and I will be the moderator of this webinar today. Our speaker today is Dr. Stephanie Bannister. She is head of customer care at Lexogen. She will guide you through the innovative solutions in the RNA-Seq workflow, which are offered by Lexogen. Before we start, I want to give you some brief information. This webinar is being recorded, so please keep your microphones muted and your cameras off at all the times. If you have any questions throughout the webinar, you can type them into the Q&A box, which you can find in the bottom of your Zoom window. We will address these questions in the Q&A session after the talk by Dr. Bannister. You can also type in questions in Japanese language if you feel more comfortable with that. We will translate these questions for Dr. Bannister and we will answer them in English. Also, an additional piece of information, during this webinar, we will ask you some short questions in little polls. Those will only take about 15 seconds and will help us to understand your needs better. So we also want to thank you in advance for helping us and for your participation. And now, without further ado, I will hand over to my colleague, Stephanie. Stephanie, up to you.
Thank you, Martina. Minasan konnichiwa. Hello and welcome from my side. My name is Stephanie Bannister and I am Head of Customer Care at Lexogen. And it is my privilege to present this webinar today, where we will highlight for you Lexogen's innovative solutions in the RNA-Seq workflow. To begin with, let me tell you a bit about Lexogen and our history. Lexogen is a biotechnology company founded in 2007 in Austria, and we are dedicated to developing technologies for complete transcriptome sequencing. Our Vienna headquarters houses our development, production, and commercialization teams, as well as our RNA sequencing services. In 2014, our USA subsidiary Lexogen Inc. was launched to serve our US customers with logistics, sales, and technical support. Of course, we also have a worldwide distributor network serving customers globally. And in Japan, our distributor partner is, of course, CosmoBio. At Lexogen, we offer you kits and solutions for each major step of the RNA-Seq workflow, right through from sampling to RNA preparation and extraction, NGS library preparation, and NGS data analysis. For all of these products, we also offer RNA sequencing services where we do the work for you. All you need to do is send us your samples and we do the rest. We'll highlight 4 key products today: QuantSeq, CORALL, RiboCop, and Trapper. But if you are interested in any of our other products or services, please be sure to reach out to CosmoBio anytime. At Lexogen, our mission is to empower our customers with innovative, top-quality RNA analysis solutions and support. As scientists ourselves, we are dedicated to offering innovative technologies that support researchers like you to perform state-of-the-art science. Today, I will highlight some of our customers' research questions and how our technologies have helped them to publish their results. We will focus on 3 core challenges: gene expression profiling, whole transcriptome RNA-Seq, and small RNA isolation and sequencing. Before we begin, we'd like to ask you the first of our poll questions. The first question is, what is your major challenge when setting up an RNA-Seq experiment? You will see a pop-up window on your screen. Please select the most appropriate option. Thank you for your feedback. Let's continue. So as biologists, we are all driven to understand how biological processes are controlled on a molecular level. And in many cases, our first challenge is to try and understand the changes in expression levels of protein coding genes. To do this well, we need a fast and easy way to assess differential gene expression in any sample. Some examples where this is required include characterizing newly generated cell clones, for example, from CRISPR screens, analyzing transcriptomic changes that happen as a result of viral infection or vaccination, screening for compounds to treat diseases such as dementia, and investigating causes of cancer using transcriptional profiling of patient samples. What all of these studies have in common is that QuantSeq, Lexogen's 3' mRNA-Seq technology, has facilitated their publication. You can see here that we have a number of webinars also associated with these studies. This is denoted by the small screen icon, and we will be providing you with these slides at the end of the presentation, and you can click these icons to watch the webinar and click the links to learn more from the publications. We will have a closer look into one of these studies in just a moment, but first let me give you a short introduction into QuantSeq and why it is so beneficial for gene expression projects. The core of the QuantSeq technology is 3' expression profiling. When you only want to know the expression level of a gene, you don't actually need to cover the entire sequence of the gene with reads in order to get expression levels. It's sufficient to only generate one fragment from each transcript and count these fragments as a way to record gene levels. This is what the QuantSeq technology does. QuantSeq generates one read at the 3' end of each transcript, which can then be counted. This gives you most efficient use of sequencing space where every read counts. It also enables a high level of sample multiplexing and enables a more straightforward data analysis by counting only fragments per gene. These factors combined make QuantSeq extremely cost efficient. Furthermore, QuantSeq is a fast and robust protocol. It consists of only 5 major steps and requires no pre-processing such as rRNA depletion or mRNA purification. Similarly, it doesn't include any RNA fragmentation. This streamlining of the protocol means that you save time and also consumables. So you may be asking, how does QuantSeq actually work to generate only one 3' fragment per transcript? QuantSeq starts by using an oligo-DT primer to prime reverse transcription of first-strand cDNA. The next step, we remove the RNA template, which allows second-strand priming to start from random primers. Only the last random primer that binds at the 3' end of the cDNA will generate a double-stranded cDNA library that contains both of the partial sequencing adapters that are added during priming stages. This library is then amplified by PCR, where indices can be included for sample multiplexing. The whole procedure takes only 4.5 hours and includes only 2 purification steps. Then the libraries are ready for quality control and sequencing. So now that I've shown you how QuantSeq and 3' expression profiling work, let's take a look at how this was used by our customers in Belgium to study vaccine responses. It's well known, especially in days like today with COVID, that people are vaccinated against diseases, but not everyone will respond in the same way. This group wanted to study how hepatitis B vaccine responses differ, and they wanted to know how these were controlled on a molecular level. They set up a study containing 34 non-vaccinated individuals and vaccinated them on day 0 and day 30 to see what molecular pathways were responsible. They then took blood from these individuals on days 0, 3, and 7, and also measured hepatitis B antibodies on days 0, 60, and 180, and 365 post-vaccination. They were able to classify from the antibody titers 3 groups of responses: non-responders, low responders, and strong responders. When they correlated the results of the transcriptional profiling with the response categories, what they could see is that responders show strong differential expression early after vaccination, around 3 days, while non-responders show a delayed response to vaccination with differential expression increasing only after 7 days. They also saw that the strongest differences between responders and non-responders were found on day 0 before vaccination began. This told them that the individuals were actually preset to be strong or low responders, and they could use this data to further predict vaccine responsiveness. Another challenge facing many researchers in terms of gene expression is to study the biological mechanisms of disease such as cancer using tissues from biobank resources. These tissues are often very old, even kept up to longer than 8 or 10 years, and they're usually derived from formalin-fixed paraffin-embedded tissues. The RNA that can be isolated from these tissues is usually very heavily degraded, where we cannot even get a reliable RIN score. Instead, RNA quality in these cases are measured by the DV200 value, which reports on the percentage of nucleotide RNAs greater than 200 nucleotides in length. So the lower the DV200 percentage, the more degraded the sample will be. These samples are very difficult for handling, and also in terms of getting good quality gene expression data using standard RNA sequencing approaches. However, QuantSeq performs really well on these samples because we only need to generate one fragment from the 3' end per transcript. Here I'm showing you the results from one study where they could detect even up to 18,000 genes per sample in FFPE samples, and showed a great correlation between fresh frozen and FFPE samples from tissue-matched patients. Why does QuantSeq work so well for FFPE samples? The answer is because QuantSeq is a 3' method. As you remember, the fragments for RNA sequencing are generated from the poly A tail into the 3' end of the transcript. And only one fragment per transcript at the 3' end is required to count the expression level of the gene. In RNA derived from FFPE samples, which is highly degraded, you will still usually have a short poly A tail present that is sufficient to bind the oligo-DT primer in QuantSeq. This way, you can still generate 3' fragments, which will only be a little bit shorter than for intact RNA. And these will still give you enough length for sequencing and robust gene expression analysis. So QuantSeq has been the method of choice now for several FFPE patient sample profiling studies. Here I'm highlighting just 3 of them. This one that is highlighted with a webinar is the one that I just mentioned. We also have a second study that is published recently on profiling FFPE samples from uterine leiomyomas. And we have a second webinar looking at the comparison of transcriptome profiles from fresh frozen or FFPE patient samples in breast cancer. And this is particularly interesting because the authors compared QuantSeq to a range of other methods and showed how well QuantSeq really performs on this sample type. So to summarize, QuantSeq is Lexogen's most cost-efficient differential gene expression method using 3' mRNA-Seq technology. You can save sequencing costs by high-level multiplexing. It's fast, easy, and robust, and works with various species, a large range of input amounts, and is ideal for FFPE samples. Before we move on to our next case study, we'd like to pause for now for our second poll question. This time we would like to understand how many samples do you usually process in one RNA-Seq experiment? I'll give you a few seconds to choose your answer. Thank you for your feedback. Let's continue. So far, I've described the use of QuantSeq for everyday applications in the lab with usually relatively small project sizes, up to several hundred samples. In this case, each sample can be feasibly processed individually, and each sample has a unique index that's added during PCR to enable identification after sequencing. But what if you want to sequence hundreds of samples or thousands of samples for gene expression levels? Manual handling is going to be tricky. This can be relevant for projects like drug compound screening, CRISPR guide RNA, and RNAi screening studies. For this purpose, Lexogen offers 2 solutions: QuantSeq automation on liquid handlers and QuantSeq-Pool. In the biotech industry and also for sequencing facilities, automation is a key factor to reduce variability and increase throughput. QuantSeq can be automated on a range of liquid handling instruments that you can see listed here. Many of these are used by our customers worldwide, and some of these setups are already featured in publications. From one of these publications, we also have a great webinar. This is from Michael Wilson's lab at the Sick Kids Institute in Toronto. But what if you don't have access to automation or you want to process multiple plates of 96 samples in parallel very quickly? In this case, our second option, the QuantSeq-Pool Kit, provides a solution. As we saw earlier for QuantSeq, you process each sample individually through 5 core steps of the workflow. QuantSeq-Pool uses the same workflow as QuantSeq 3' mRNA-Seq. However, at the first step, each sample gets a unique sample barcode attached by the oligo-DT primer. This means that after the reverse transcription, you can pool all 96 samples into a single reaction. The rest of the protocol is then carried out in a single tube. This allows you to easily scale up the number of reactions processed, reduces time and consumables. So the benefits of QuantSeq-Pool are many, including namely that you save time in QC and normalization, as well as the preparation of the libraries and sequencing run. You save consumables up to 10 times less than regular QuantSeq, and you can decrease the technical variability by pooling the samples early in the process. Combining QuantSeq-Pool with the available 384 unique dual indices that we have from Lexogen, you can sufficiently exploit the massive NovaSeq capacities that exist, sequencing up to 36,000 samples at once. So to summarize, QuantSeq-Pool is designed for high throughput expression profiling screens and is the most cost-efficient method for massive scale multiplexing. Benefits include huge multiplexing up to 36,000 samples, saving 90% on lab consumables and sequencing costs, and enabling you to process hundreds of samples in parallel in a single day. So now it's time for our third and final poll question. Please take a few seconds to share with us what is your key decision maker when choosing a library prep kit. Please select your option from the list. If you don't find something that fits for you, you can select others. Thank you for your feedback. Let's continue. So we have seen now how QuantSeq is an efficient technology using 3' RNA-Seq to profile expression levels. As we know, however, transcriptomes are complex and genes consist of multiple exons and introns and regulatory sequences, and you may need to look at more than just gene expression. For example, you may need to do isoform discovery, understand alternative splicing activity, and look at SNP detection. For these approaches, you need a whole transcriptome RNA-Seq library prep method. Lexogen's solution for whole transcriptome library prep is CORALL. CORALL can be used for total RNA sequencing or mRNA sequencing, and it works with any sample, including low input and FFPE samples. It's an all-in-one solution which includes unique molecular identifiers and unique dual indexing. And like our other protocol, QuantSeq is a fast and streamlined protocol that is also compatible with automation. Here you can see some of the performance data from CORALL RNA-Seq libraries, which we prepared with ribodepletion. You can appreciate that we see robust gene detection up to around 25,000 genes, even with inputs as low as 1 nanogram of total RNA. In addition, compared to competitors, we can detect 96% of genes that other competitors detect, as you see here in this Venn diagram. I will mention that this is a highly stringent filtering, looking at genes that have an expression level of more than 10 C.
So what types of research questions? Excuse me.
I hope you can now hear me again. Sorry for this technical hiccup. So let's continue. What types of research questions are researchers using to solve, using CORALL to solve in their labs? The applications are very broad, including validation of drug candidates, multi-omics analyses for validating new small molecule compounds in drug discovery, human and viral RNA studies. For example, this can include looking at SARS-CoV-2 effects on the nervous system in post-mortem FFPE stored brain biopsies, and sequencing ribosomal RNAs and total RNA in parallel in Arabidopsis. So all of these researchers chose to use CORALL because they needed to have a more in-depth analysis of the transcriptome. And what does more in-depth mean? It means that we wanted to look for isoforms or to look for the complete sequences of protein coding genes. In these examples, the researchers used mRNA selection so that they could look at the protein coding fraction specifically. However, what if your research is not only focused on protein coding genes? Selecting all protein coding genes will restrict the analysis to poly-A containing transcripts, but many researchers aim at looking at having a more neutral approach and looking at long mRNAs, no matter if they're polyadenylated or not. This can include looking at validation of anti-cancer activity of newly developed compounds, clustering tumor samples into subgroups based on transcriptomics data, and expression analysis of long non-coding and coding RNAs together. For these studies, our customers combined the use of CORALL and our Depletion Kit, RiboCop. RiboCop itself is a highly flexible kit that can also be used in combination with most other whole transcriptome library prep kits available. RiboCop can handle a wide input range from 1 nanogram to 1 microgram of total RNA with high efficiency. It uses a magnetic bead-based workflow and highly specific probes to minimize RNA loss. We have specific protocols for human, mouse, and rat, and bacterial ribosomal RNA depletion, and a solution that combines mammalian ribosomal depletion with globin depletion. So RiboCop works, as I mentioned, by hybridization of total RNA to ribosomal depletion probes. It uses state-of-the-art technology of probe design for best efficiency and minimal off-target effects. We then use a magnetic bead-based approach to capture and remove the ribosomal probe captured sequences. This makes it fast, easy, and also automation friendly. RiboCop shows excellent depletion efficiency across species. Here we show the results for humans, mouse, and rat using our HMR version 2 kit. This will bring you down to even down to 0.1% of remaining ribosomal RNA, which can be species dependent. And we would like to note that this is outperforming other kits on the market at the present time. Here you can see how the RiboCop bacteria kits perform for 3 different species of bacteria: E. coli, Pseudomonas aeruginosa, and Bacillus subtilis. Depending on the species and the input, you can also deplete down to 0.1% of remaining ribosomal RNA. Lexogen offers 3 different RiboCop kits for bacteria to suit the type of bacteria of interest. These depend on the gram status, so we have gram-negative and gram-positive specific kits, and a meta kit which can deplete complex mixed bacterial populations, for example, that might exist in microbiome or environmental samples. These results I show here are also from the meta kit, so you can use this kit also for regular bacterial species as well. But many researchers want to go beyond the routine analysis of the transcriptome. They want to understand more than long protein coding or non-coding transcripts, and how these transcription levels are regulated. This can be largely influenced by the action of regulatory small RNAs. These include microRNAs, siRNAs, small nuclear RNAs, and piRNAs. The usual starting point for small RNA analysis is to do a global analysis of small RNA expression, and for this, small RNA sequencing fills the need. However, the existing challenge in the field of small RNA research is how to prepare the small RNA fraction for meaningful insights. Ideally, you want to use an RNA extraction method that is specific for your small RNAs of interest. The most known methods to extract or select small RNA for NGS are depicted here. Firstly, column-based methods are easy to use, but not very specific, since they apply a general size cutoff of around 200 nucleotides and simply elute all RNAs smaller than this. So with this method, you will get a lot of small RNAs, but you will also get degraded RNA fragments alongside. Another option is to use gel purification to increase the specificity by selecting a narrower size range. With this method, you will also get some degraded RNA fragment carryover, but it is a lot more difficult and does not work well with low amounts of input RNA. There's also a high risk of yield loss. A third approach is to use co-immunoprecipitation of the small RNAs of interest bound to their respective protein complexes, which usually involve Argonaute proteins. For this, you need specific antibodies that usually only work with one given species, and the co-IP conditions can be time-consuming to optimize. As a solution for this problem, Lexogen now offers TRAPPER. TRAPPER is a small RNA isolation kit which provides specificity and sensitivity along with speed and ease of use. It's a column-based method that is also fast and straightforward that only isolates small RNAs that are bound to Argonaute proteins. Let me now show you a bit more about how TRAPPER works. TRAPPER stands for Trans-Kingdom Rapid Affordable Purification of Risks. The first step is to lyse cells using the TRAPPER lysis buffer. This will keep the risks, the Argonaute proteins bound to small RNAs intact, and this protects the small RNAs from degradation. This lysate is then loaded onto the TRAPPER column, which uses a principle of ion exchange chromatography to trap RNA and DNA on the column and allow the Argonaute risk protein complexes to flow through. You then elute the risks containing the small RNAs of interest into the eluate, leaving the RNA and DNA that you're not interested in bound to the column. Even though TRAPPER was only released in summer 2020, there are already some proven examples of how it can be transformative for small RNA research. For example, enriching the function or silencing small RNAs for all organisms, distinction between active and free-floating small RNAs, and profiling small RNAs to study allelic expression regulation in plants. We can recommend the use of TRAPPER together with Lexogen's small RNA-Seq library prep kit, but TRAPPER can be used together with any other downstream small RNA process, including qPCR, or with other small RNA library prep kits you might have established. So to summarize, TRAPPER is the only kit on the market that is able to specifically isolate functional small RNAs from any species. You save, excuse me, you save sequencing costs by focusing sequencing depth only on the small RNAs of interest, which gives you data on physiologically relevant RNA species. And the workflow is fast and gel-free, which enables you even to work on challenging samples like plasma. So today I have shown you some examples of how QuantSeq, CORALL, RiboCop, and TRAPPER are helping scientists to reach their research goals. However, what if you don't have the time or the expertise to set up NGS methods in your own lab? In this case, at Lexogen, we also offer sequencing services. These utilize all of our catalog solutions. Our services facility is located in Vienna, but you can ship us your samples, frozen or as extracted RNA, and we will run the whole process for you, including data analysis. Our service offering is unique in that we provide personalized treatment with individual consultation for every project and custom tailor our approach to meet your needs. You will have peace of mind with our project management and technical expertise. And because we are close to the developers of all of our products, if we need to modify something to suit your specific experiment, we can do that quickly and effectively. You can rely on high quality results thanks to our competence to handle samples even outside of standard specifications. So please get in touch with us if you would like more information on our specific services for any of our products. With this brings me to the end of our webinar. If you would like any more information about Lexogen services, you can explore these on our website, or even better, reach out to our partners at CosmoBio for more information. With that, I would like to say, go seichou arigatou gozaimashita. Thank you for your attention, and I'll be happy to answer your questions.
Thank you, Stephanie, for this amazing presentation. So we have received a few questions in the Q&A box. I will start with the most recent one about Trapper. Does Trapper extract RNA, or does it need to be coupled with an extraction kit?
Thanks, that's a very good question. Our Trapper kit includes everything that you need for the RNA extraction after the column processing, so you don't need an extra kit.
Thank you, Stephanie. Then going back in our line, next question is about CORALL. Is CORALL also recommended with FFPE samples, and what is the recommended input amount for those samples?
The answer is yes. So CORALL also works really well with FFPE samples, and you can use any input from 1 nanogram to 1 microgram. We recommend to use RiboCop for ribodepletion before starting CORALL for FFPE samples. This will give you the best results across your gene and best gene coverage.
Thank you, Steph. That was clear. So you mentioned RiboCop. There's a question about RiboCop. Can RiboCop be used to deplete ribosomal RNA from degraded RNA samples?
Yes, so RiboCop is our main recommendation when you're working with degraded samples, even if they're not FFPE. When the RNA is more fragmented, it is much better to do a ribosomal depletion before doing CORALL, so that you can still get rid of the ribosomal RNA, but you won't bias the sequencing to the 3' end, which would happen if you would use an mRNA procedure.
Thank you, Steph. Another question, are all the presented kits compatible with Illumina sequencing?
Oh yes, I think we did not mention this, but all of our portfolio are compatible with Illumina sequencing. The Trapper kit is actually working upstream of any library preparation, so this is compatible potentially also with other platforms, depending on which library prep kit you use. But QuantSeq and CORALL and RiboCop are all compatible with Illumina.
Okay, thank you, Stephanie. Now there's a question about QuantSeq coming in. What is the recommended input range for QuantSeq library prep?
Yes, good question. So it's very broad. You can start from 1 nanogram of total RNA up to 500 nanograms of total RNA. And I will add that the protocol does also work with lower inputs, and please get in touch with us if you would like to work on those. We have a few quick protocol recommendations.
Okay, thank you, Stephanie. So those are the questions so far. As a reminder to the audience, you can also type in the question in Japanese language. We have colleagues here to translate those. I think we'll give you a few more seconds if you have another question. And in any other case, you can always contact our colleagues at CosmoBio if you have questions about our products. There's a question coming in about QuantSeq. Does QuantSeq enable us to analyze alternative polyA site usage in FFPE samples?
So we have an alternative version of QuantSeq called QuantSeq Reverse, which is really well designed for studying alternative poly-A sites. QuantSeq Reverse works with the same workflow that I've shown you in the presentation, but we swap the orientation of the sequencing adapters and use a custom sequencing primer for the sequencing run. And what this lets you do is start to read the sequence that begins at the very end of the transcript back into the transcript itself. And this way you can see the poly-A signals at the end of the transcript that show you where the termination of transcription will end. So if you use QuantSeq Rev, you'll be able to map these alternative poly-A sites. In principle, this can also be used for FFPE samples, but it is going to give you a little bit more limited information because the sample to start with will be very degraded. So you probably wouldn't get as good resolution as you would with an intact sample, but you will be able to detect some alternative 3' end sites.
Okay, thank you for this question and this answer. So I think there's nothing more coming in for now. Then with that, I would like to close this webinar. Thank Dr. Stephanie Bannister again for this awesome presentation. And thank you for your attention. And if you have any questions left, please contact Cosmo Bio or our support at lexogen.com team. Thank you, everybody, and have a nice evening or a nice day. Bye-bye.
Thank you. Bye-bye.